Okay, I think we'll get started. Welcome, everybody. Welcome to the fourth and final evening of the 2019 Brain School as part of Brain Awareness Week. We're very pleased on behalf of the Fralin Biomedical Research Institute to be able to bring this to the community, and really glad to see the great crowds we've had every evening. For tonight's topic, I certainly don't see any aging bodies out there, and I guess there aren't any aging brains either to go along with that, but clearly we're all interested in that for somebody else. Firstly, I want to say that, I think it's been a great week. The topic tonight is the CAP, and it seems appropriate that we'll hear about what happens to brains as they move on in life. And we'll once again follow the same format where we have a scientific research presentation and then a presentation more on the clinical side. And also I want to make sure to mention that I hope everybody who attended all 4 evenings picked up their certificate, their graduation certificate out front. Don't think you can run out now with that certificate. It will void immediately through disappearing ink if you don't sit through tonight's lecture. So that's all part of it. And for all of you who have attended all 4 of the events and received a certificate, would you please stand? Wait, hold your applause. Okay. Wait, stay up, stand up, please. By the authority and powers vested in me, I hereby award you Brain School graduate. Go forth, congratulations. Okay, I'm told to keep talking, there's a little computer glitch. So let's talk about rattlesnake brains again. Are we good?
Yeah, I'm just going to transfer. He wasn't showing me the brain.
It's what?
He wasn't showing me the brain.
Oh.
I'd like to see the brain of the animal that eluded the rattlesnake.
The animal got away. That was the kangaroo rat.
I'd like to see his brain.
Yeah, the gentleman brings up a good point. Everybody remember the kangaroo rat twisting in midair and getting away from the ballistic rattlesnake strike? Now that's a brain. You're absolutely right. Incredible fast response. Are you good now?
Double see the movie's playing.
You have to check it out. You're good? You need to check it out? Or you need to check it out? Or everybody's going to see it if you do it.
Yeah.
I would say forget it. Just wait till you get up against it. Okay, so after that small delay, we will get started. So tonight's speakers include Dr. Greg Valdez, who's an associate professor at the Fralin Biomedical Research Institute and a renowned researcher on brain aging, both healthy aging and neurodegenerative diseases and the underlying biology and mechanisms involved. So you'll hear from Dr. Valdez first. And then he will be followed by Dr. Brian Unwin. Dr. Unwin is the Chief of Geriatric and Palliative Medicine at Carilion Clinic at Virginia Tech Carilion School of Medicine, who will share with us some of the aspects of what happens in the clinical setting and day-to-day life with any of us or patients as our brains may age and as certain neurodegenerative processes such as dementia set in. So it should be, once again, a great balance of the science and the medicine. We will follow the same format. Dr. Valdez will speak for 25 to 30 minutes, then Dr. Unwin will speak, and then we'll have plenty of time for questions at the end. So without further ado, it's my great pleasure to introduce Dr. Greg Valdez. Greg.
Thank you, Mike. I love how Mike is so assertive. As we were having that little computer glitch, he said, "Go sit down so I can introduce you." And on command, I did. He's the boss. It's a pleasure to be here, and haven't had the fortune to come to every one of these talks this time. Participated in previous ones. I attended almost every one of the previous talks, Brain Week talks in previous years. But this year I have heard that it has been a resound success, and I'm very happy to hear that the community is willing to come in here and hear about what we're doing and what a lot of our colleagues at Carilion and also in the main campus are doing. So what I'm going to do is give you a couple of big pictures, one about us as human beings, what we need to do to ensure that we maximize just our natural abilities, and the other aspect, which is really what my research is about, is trying to figure out whether the machineries that we have, our cells can be modified in any way possible to slow down all the ravages that come with aging. Not just normal aging, but also that comes with diseases of aging, like ALS and Alzheimer's disease. Now, I think all of you are going to agree with me that this is not aging. This is actually one of my colleagues that spoke here a couple of days ago, Mike Fox, when he was a little kid, and this is his wife, Amy. I did not tell him that I was going to show these pictures, but at this point here, he probably began the normal aging process. He probably was around 26 years of age, and certainly around this age now, in his ripe 30s, he's already in the normal aging process. And obviously, this is a computer modified version of himself where he has aged a lot more. Mike actually is fighting pretty successfully, I think, aging with all the exercises that he does and all the social activity, and certainly being a professor and his ability to communicate and interact with so many different individuals in so many different ways. Very, very important. So this is no aging. But aging happens, and I don't have to tell you guys a whole lot about the statistics. We know that as a nation, as a world, we are aging. By 2035, individuals that are 65 years or older are going to outnumber individuals that are 18 years or younger. So these are the 65 years and older, and this here are the 18 years and younger. So this is going to be an aged population, aged nation for every practical purpose. So we need to do something about it, right? Because with aging, there's a lot of issues that come, and we need to find ways to modify them. We're not very good at modifying the problems that come with aging simply because we're human and we get lazy. We also like too many things that are not very good for us to fight aging. One of the biggest problems that we know happen with aging is that we increase our body mass index. We eat too much, we become more sedentary, and that is, if anything, much more closely associated than anything with decreased longevity. There are a lot of other issues that come with aging, but this is, if anything, the biggest issue that we have as a nation, the obesity problem that we have. So we have to fight it. Obviously, this is a U-curve shape, and so if you're too skinny, you also have a bit of a problem. You need to have enough calories, sufficient calories for your system, sufficient fuels for your cars to be able to go, your brain, your heart, and every single processing machinery of your body. But this is exactly where we want to be if we want to increase our lifespan. Now, physical activity increases our lifespan. We know that. Simply exercising for 30 hours a day, a week and not a day, that's a little bit much, 30 hours a day. I'm an alien, but not that kind of alien. There are 30 hours a week can extend your lifespan by at least 5 years. So that's quite significant when you think about it. It's not just a lifespan, but it's your health span. It's your ability to communicate with your loved ones, your ability to participate in many of the activities that you have participated with your loved ones for so many years, your kids and your grandkids. So physical activity is something that we know increases the lifespan, increases our ability to connect different processes. So just a little bit of exercise, aerobic exercise, nothing too extraneous, increases brain volume in many different places. The white matter of the cortex, the anterior circular gyrus, for example, the left temporal cortex, all of these areas are increased simply by a little bit of aerobic exercise every single week. And these are places within the brain that are really important for speech, for making the right type of decisions, the go and no-go type of decisions that we have to make on a daily basis in our lives. Exercise, just a little bit of exercise, not only increases the volume of different areas of the brain, but it also sustains them. So individuals, for example, in this experiment here, where individuals were asked to do was to do some leisure activity for about a year. And the brains were scanned using an fMRI to look at their activity. And then those individuals were asked to stop doing those activity. And then they were tracked for another 2 years here, individuals that were active and individuals that were sedentary. And what happened is, as you can see here, is that the brain of those individuals who were active show much higher brain activity. Not only were their brains more active, remain more active even after they stopped exercising, but their ability to commence different motor function and to commence them at in a speedy fashion was much better just by 1 year of exercising. Now, the famous writer, Gabriel Garcia Marquez, the Colombian writer, had this quote in his book, Love in the Time of Cholera. All age comes with the first fall and death with the second. And that is really, really important. It's really important, especially in this age that we live in, because falls are the biggest contributor of health decline in our population. You fall, you become more sedentary, you get depressed, you stop talking to your loved ones, your friends, and then all those positive and others that Brian will show you later, positive effects of exercise, not just physical exercise, but cognitive activity, type of cognitive exercise, are gone. Right? So your health span decreases quite dramatically. Your lifespan also decreases. Now, there are populations around the world that researchers for many, many years have tracked. Aging is not a continuum across every single individual that is exactly the same. It varies depending on the place of the world that you look at and the kind of the activity that the individual is engaged in. There are three, generally speaking, areas in the world: in Sardinia, Italy, Loma Linda, California, and Okinawa, Japan, where individuals in that part of the world live longer. They live much longer on average than anybody else in any other region of the world. And that is because they're physically active, they eat a better diet. Certainly they are full of antioxidants. That doesn't have to necessarily mean that that is the only reason that they live longer. But they do have much stronger social networks. They get up every morning and they do their chores in their patios, and they communicate with each other, have wine together. So all of this is important. It's not just the physical activity of the diet, but it's also the social interaction. And this is why I'm so happy to see so many people here. And hopefully a lot of you will continue to talk about the research that we do, but also what each of you does on a regular basis during your lifespans. Now, there are individuals that we know of that are super active on a regular basis, and he's probably going to bend that whole lifespan curve that I've shown you. One is Mike Fralin, obviously. You know, these individuals here are saying, "Go, Super Mike." He's always doing something at work, physically, but he's also very socially engaged with all of us, and certainly with his family. So I'm sure he's going to bend that curve by quite a bit. He's probably going to outpace this lady here, Madame Joan Comment, who's the longest living individual on record that I know of. I think she lived up to about 123 years of age. She lived in the southern part of France. She was active until the very last day that she passed away. She biked to the market on a regular basis, had a lot of friends. And the story goes that when she was 90 years of age, a businessman approached her. She had a beautiful apartment in a beautiful location, perfect location in the southern part of France. And this business guy approached her and said, "Hey, I'd like to buy your property." And she said, "Well, sure, I'll sell it to you if you let me stay in my apartment until I die. And I'll even give you a discount," okay? And he fell for it. He said, "Sure, I'll buy you, you know." So that was 90 years of age. You do the math. 30 plus years later, she's still alive and she's living rent-free and pocketed that money. And without interest, she was a lot richer. So she had not just the physical abilities, but she had the cognitive ability, the ability to process that information really quickly and figure out that, no, I can actually take you for a ride rather than the other way around. So again, very, very important to stay engaged in so many different facets of your life, not just physically, but cognitively. Now, the brain ages, we know that, irrespective of what we do. Aging is something that we simply cannot stop, not at this moment. We can't. Maybe someday. I doubt that we can stop all the issues that come with aging. But one of the things that happens that we know is the brain shrinks. Obviously, cardiovascular decreases, there's diabetes, there's a lot of issues that come with aging, in addition to the age-related neurological diseases. As a neurobiologist, that's what I'm going to focus on mostly. We know that with brain aging, Alzheimer's disease, Parkinson's disease, gait disorders, amyotrophic lateral sclerosis, a lot of diseases that are associated with aging. What does that mean? What it means is that the cells have the ability to keep at a distance whatever toxic substance known, oftentimes a mutation in the gene, sometimes unknown, it could be manganese or some kind of other factor in the environment, can keep it at bay from killing it, from causing any real damage to its normal function. But eventually, it can't do that anymore, right? It can't, or it simply can't fix those damages that those mutations or those toxic substances are causing. It can't fix them fast enough. And so they give in, they die, they start to die, and that's when these diseases come about. They age-related because the neurons, the cells that are aged now fail to maintain themselves as well as possible, as they used to when they're younger. So what happens with aging? The brain shrinks, we do know that. There are a lot of technology, fortunately, that have come about and that continue to come about that allows us to pry into the brain. Now, with humans, you don't want to really pry into the brain of a normally functional individual without any disease, unless you really have to, right? Because they have a disease or need some kind of intervention. So there are technologies such as fMRI that allows us to generally just look at how the brain functions, how it's constructed. It gives us a map of not just the size of the brain, but what regions of the brain, as I showed you before, those red areas or blue areas of the brain, how they're changing, whether they're active, whether there's a little bit more of an area of the brain versus another. So this is one of those modalities, one form of MRI or fMRI, where we can actually look at the gray matter of the brain, and that is where the cells are. That's where the biochemical center of cells, the cell body, as we call them, are. And then the white matter, where the axons and dendrites, where they make connections with each other, are for the most part. So we can look at those in a human. Obviously, this is looking very broadly. This is looking at the forest and just seeing what changes somewhere in the forest have happened. Not very, very specific. But nevertheless, that gives us quite a bit of information. It tells us that the gray matter of the brain doesn't really change a whole lot with increasing age. So your cells are there. Your neurons, your glial cells, most of them are there. You haven't really lost a whole lot of your gray matter. But what does change is the white matter, where the axons and dendrites, those areas that the neurons utilize to talk to each other, they're disappearing. There's less of them. So that's quite powerful. Okay? With additional technology, we can look into the brain. Now, this is a very old technology by the famous father of neuroscience, Ramón y Cajal, Santiago Ramón y Cajal, who was a Spanish neurobiologist in the 19th century. He stained with a technique called the Golgi technique, the brain, and it allowed him to see just a few neurons. It's just one of these techniques that somehow just labels a few neurons here and there. And by doing so, he can see that these are the cell bodies, and then these are the extensions that we call dendrites or we call axons in a neuron. In a human being, we can look at them once the individual has passed away. We do not want to take someone's brain away. Or we can look at these neurons if we have to remove a portion of your brain because of Parkinson's or because epileptic seizures. Only in those cases can we utilize, we consent that kind of tissue. And that is pretty finite. That's just the end point. With human tissue, we can't look at the continuum of aging to really know where the balance tips from normal to deleterious. From normal, from being able to slow down the ravages of aging or a mutation that is trying to kill a neuron, to not being able to do so. Right? And this is where animal models come in. Animal models allow us, better for worse, to do a lot of things that we simply cannot do with humans, and that is look at neurons, glial cells, and other tissues throughout the entire lifespan. Two mouse models that I use, not here, one of them, monkeys. This is with a collaboration with the National Institute of Aging. One that I use here regularly, mice in the lab. These animals live a lot shorter lives than we do. And We are allowed to do research on these animals. Mice live on average about just over 2 years. Their maximum lifespan is just over 3 years, and monkeys can live on average 28 years. These are rhesus monkeys, so macaca mulatta monkeys, and a maximum lifespan of 40 years. This is still a lot faster than assessing the lifespan of a human being and changes throughout the lifespan. Most importantly, we are allowed to do certain things that we simply would never do with any of us. And one of the things that has come about that we can do, and with the advent of a lot of technological advances, is we can look at neurons in ways that was not possible before. So before, we simply looked at all the neurons in a static image from Ramon y Cajal from the end of the 19th century. Now we can look at those neurons and look at subpopulations and look at cells around them, multicolor them. These are neurons from the cerebellum of a mouse. This is actually a transgenic mouse that a guy that I worked for, Harvard, generated. It allows us to do this, and so we can look at each one of them and ask, why is this in this position versus this one here? Right? We can obtain more information now that we simply could not before. Certainly we can do a lot more than that and just look at them and see their position and whether they're there or not. We can record from them, something that Freelander does on a regular basis. Listen to them talk to each other and see how their conversation changes in disease context or during aging or traumatic brain injuries. That's something that we can do. We can manipulate their genetics. We can put in a mutation that causes a disease in human, and then we can take it out and see if we can reverse the damages that mutation has caused in those neurons, or see how those damages come about. So there's a lot of stuff that we can do with animals that we simply cannot do with humans. So that's my pitch for doing this, and let's see if we can play this movie. Can you? No, you have to click on it. Sorry, we had a computer glitch here. This was supposed to be played from my. There we go. So if you just go back to these images here, these are static images, just like the images that I showed you previously with the Golgi stain. If you look at this now, with these new technologies that we have, it's not just genetics and using the natural resources of life, using this fluorescence proteins that allows us to just put them into a cell and let them fluoresce and allow us to look at them. We can now use specialized optics that we can look right into the brain of a live animal and look at every neuron. These are a couple of microns, the size of a couple of microns, and look at them and see how they're talking while the animal is making decisions. So this is a mouse that was put in a new environment. So it's just trying to figure out where it is. Different cage, the context was a little bit different, the texture was a little bit different, and they're looking specifically in the CA1 region of the hippocampus. You guys don't know what that is, but the CA1 region of the hippocampus is involved in spatial learning. So it basically tells us and tells animals what it is, where we are. And it allows us to remember those locations and specifically the parameters of that location. That is also the place which is first affecting Alzheimer's disease, okay? In humans, but also in animals. And so what I did here was simply look at what neurons are talking, activated. And this is the image here. This is the animal moving around, trying to figure out now its new environment, that these neurons are firing. They're basically talking, communicating with each other, and decoding this new environment that they're in, and making a memory of this new environment. Eventually, this animal will settle down, know that this is home, and he knows exactly every corner of this place, and also the texture of the place. Okay? So very, very important. This may seem insignificant, but very important then for looking at these same neurons in the same region of the brain that is affected with Alzheimer's disease, and see if it can form those type of memories, see if they can talk the same way in a normal situation like it is here. Now there are other examples of things that we can do. This is a much higher magnification of neurons just responding to visual changes. Just different bars, and there are certain neurons that are talking constantly when specific type of visual information is presented to them. Again, There's a lot of computational power that we can delineate from looking at those neurons. We know where they are, we know their location, we can go in and ask, what makes you so unique? What are the molecules that are in you that are not in a nearby neuron? There's a lot more that we can do, and I won't show you every single example, but here's another very powerful assay that we can do with some of the new technologies that the scientific community has been able to generate just by looking at nature, using these fluorescence proteins from a jellyfish. You can take those neurons from an animal, put them in a cultured dish, and look at them develop, redevelop their extensions, those extensions that they use to talk to each other. This is arguably the most important part of a neuron besides its nucleus, where the biochemical machinery is. They need those extensions to reform, they need to talk to each other, they don't talk to each other, and the neurons die. We have problems. And so this gives us a lot of power to see if those neurons can reform connections, can sustain connections that they have made in many different contexts, disease contexts, stress conditions, normal aging. So here's an example, another example of a monkey pyramidal neuron from at a young age, where we're looking now at one of those extensions, and we're looking at specifically a place that is called spine, where they form synapses, where they begin to talk to each other, and you're only looking at half of that bridge, okay? The side of communication. The other side is not visible. You're looking at this side here, and these two sides are supposed to be talking to each other, okay? This is from a young animal, and we can only see it because of the technology that we have, but also the genetic manipulations that we can do. This is from a young monkey, rhesus monkey, and this is from an old monkey. Many of those spines, sites of connections, of communications between neurons, are gone in the aged animal. So that begins to tell us that the white matter, the decrease in the white matter that I show you using fMRI previously in human, is likely due to loss of synapses. So this is the kind of insight that animal models can give us that we simply cannot obtain with humans. Simply cannot do that. That would be immoral, obviously. Now, there's a lot more that we can do with animal models, and this may scare you when you see a snake. Some people, especially my kids, will freak out if they see a snake, but this is actually a very, very powerful yet snake for what it possesses, but also for what it has contributed to the scientific community. In nature, there are fluorescence proteins, but there are also toxins. Toxins that bind specific molecules in our body, and yes, they can be lethal. In this case, this snake, it's a crate snake, a Taiwanese crate snake, secretes a toxin called alpha-bunker toxin. If it bites you, it paralyzes your muscle. And if there's a sufficient toxin, it will paralyze every single muscle in your body, including your diaphragm, and unfortunately, you will pass away. Now, obviously, that doesn't sound too good. But scientists realize that there has to be a molecule in muscles, or perhaps at the site of communication between muscles and neurons, that toxin, whatever it is, previously they didn't know which is exactly what the toxin looked like, is binding to, is preventing it from functioning. And biochemists went after it. They identified that toxin, generated sufficient amount of it, and behold, they found that it binds to a site, to a receptor that is called a nicotinic acetylcholine receptor at a synapse. Okay? And what it does is it prevents communication between a neuron and a muscle. Those synapses are also present in the spinal cord, they're present in the brain. We have cholinergic synapses all over our body. So now we have a way to visualize, specifically visualize those type of synapses. There's a lot of different types of synapses with neurotransmitters such as GABA or glutamate. It's a lot of different types of synapses, and they play different roles in our cognitive functioning, motor functioning. So nature provides us with a lot of tools, and this is one of them, toxins. And this is one of those toxins, alpha-bunker toxin. There's another one that we generate and other animals generate, and those are antibodies. Antibodies allows us to visualize specific molecules anywhere in our body and separate them from any other molecule. That way we can see whether those molecules, the number of the molecules, their distribution changes, but also just look generally at the structure that they populate, whether it's a synapse or some other place in our body. So why am I telling you that? Because in this case here, this is a synapse that neurons from your spinal cord with your muscles form. And this is the synapse that we all utilize to commence every single voluntary action, movement. This is a synapse that we have to have for our diaphragm, for us to breathe, diaphragm to function, our legs to be able to move. And this here is the muscle side. This is actually labeled with that toxin that I just mentioned. Just a little bit of it, not too much of it so that the animal wasn't killed. Just a little bit of it to visualize just the portion of the synapse on the muscle side. It actually prevents the muscle from listening to the neuron. On the neural side, we're looking at the very tip of the axon with an antibody. So now we have two different tools that nature has provided to us. It has taken many, many, many years, decades, to come to develop antibodies and toxins that are useful for the kind of research that we do, that nevertheless very important. This synapse here is from a young animal, 3-month-old animal. That's the equivalent of about a 20-plus individual. This synapse here is from an aged animal, from a 24-month-old mouse, the equivalent of a 75-year-old individual. You can see that structurally, with the power of these two synapses look very, very different. In my previous position, I looked at the changes that this synapse goes through as the animal ages. And what I found is that this cheese puffs type of structures and the lack of communication, which is the lack of opposition between the green and the red, the muscle and the neuron, is lacking and begins to lack actually to disappear pretty early in life before we actually show any overt motor deficits. So synapses are aging on an ongoing basis. But there's something we can do about that. Caloric restriction, with all the nutrients and all the metabolites that are needed, can slow this process. This is from an animal the same age as this one, but on a calorically restricted diet. Same age, a very old animal, 75, the equivalent of a human being, 75-year-old individual, same as this one, taking walks to McDonald's, eating a controlled diet. Okay? This one looks like the young synapse. Exercise. Exercise can also slow down aging. When you look at a synapse like this one, look at the muscle side, the nerve side, it looks aged again. You see the cheese puffs type of structure, the lack of opposition between the green and the red before exercise. In a sedentary animal, the mouse exercise, and this looks quite rejuvenated. Okay? There's very nice opposition. You have much fewer of those little cheese puffs. That means that cheese puffs really means that the synapse is disintegrating, it's falling apart. Okay? Now, with some of the technology that I showed you earlier, the live imaging, I tracked these changes in a live animal. And in fact, I saw that these changes are not just slow down, but exercise reverses those changes. Okay? Too much to show you an animal that is open and where we're doing live imaging here. But exercise can reverse aging of some of the synapses. Caloric restriction can slow down aging of synapses. All very, very important. And so why is this all so important for us human beings? Well, for some of the reasons that I stated before. There's just so much that we cannot do with ourselves that we shouldn't do anyway. We can't do a lot of the manipulations and investigations that we need to do to figure out why is this synapse so much more plastic in the presence of exercise. This is just all a means to an end. And why does it succumb to aging when it doesn't exercise? We can do that with animals. I'll show you a little bit of data to finish off, but just before I go there, I just want to have a very quick pitch to something that we discovered in my lab. As I said, this is all a mean to an end. The exercise, caloric restriction, we know that those are the two best ways to slow down aging. There's nothing better than that. Okay? Using this type of paradigm, we compared synapses that were exercise or caloric restricted versus none that were not exercise or were giving a regular diet. And we have found molecules that basically can substitute some of the effects of exercise. And one of my graduate students, Cindy Vaughn from here from Roanoke, found a molecule that makes this synapses much more plastic. Okay, that means that this synapses can remodel much faster, especially when they're allowed to exercise free will. Okay, so very, very important. So now we have a molecule, and you hear about that hopefully in the next couple of months. This discoveries come to be in the general press, but importantly in the scientific community. So very important, and what we hope it for is that cycle of discovery from animals to human continues. But here's one that I think is going to hit home even more so. You guys have heard about plaques, Alzheimer's plaques, for many, many years. Plaques are the ones, amyloid plaques are the ones that cause Alzheimer's disease. There's a lot of truth to that, but it's not all true. There's a study that was done in California where 90-plus-year-old individuals were tracked, everything from their social activity to the health activity to what they ate almost every 6 months for many, many years. At the end, those individuals agreed to donate their brains to research. And what they found is that individuals that were cognitively perfectly active and appear normal at the time of death, they had just as many plaques as individuals that succumbed to Alzheimer's or came down with Alzheimer's disease. So in essence, it kind of broke that relationship between plaques and Alzheimer's disease. It meant that plaques don't necessarily mean that you are going to have Alzheimer's disease. Okay? This research continued, and another lab went on right up here and looked at the plaques, their position within the brain, using a lot of the technologies that I showed you before, antibodies, biochemical assays, and what they found is that the location of the plaques is really what matters. The location of the plaques, but also whether the plaques contain specific type of amyloid beta. If the location of the plaque, if the plaques are located near synapses, those little protrusions that I showed you earlier, that we also call spines, if they're very close to synapses, Alzheimer's is going to happen. If they're away from synapses, the individual is likely to be cognitively fine until the time of death for other reasons. This work here and this image here is from work that was done in animals and then in cell cultures, proving that if neurons are treated with plaques or the derivatives of those plaques that cause Alzheimer's disease, they lose a lot of their synapses. So research now has been making a much tighter relationship between plaques, their location, their composition, and synapses. So what does that mean? What it means is that we need to find ways to either move the plaques someplace else or find specifically within plaques what is it that is binding to synapses to prevent those synapses from staying healthy. So I'm going to end there, and I'll be happy to take questions later on, but exercise, live a healthy lifestyle, eat well. Remember that there is now evidence that plaques are not exactly the end of it all for Alzheimer's disease. Science has given us a lot more information, a lot more precise information that are leading to new discoveries and ways potentially of treating Alzheimer's disease that may be able to circumvent simply the appearance of plaques. And with that, I'll leave it, and I guess we're taking questions later.